Every living cell constantly navigates a strict trade-off. It can dedicate its resources to multiplying, or it can fortify itself against damage. On the side of growth, we have beta-catenin. It acts as the central coactivator for the canonical WNT signaling pathway, which is responsible for driving the genetic instructions that tell a cell to divide. On the defense side is FOXO3. This is a transcription factor that operates as the cell's primary environmental sensor, closely monitored and regulated by the IGF-1, PI3K, and AKT signaling network. In a stable, nutrient-dense environment, growth is prioritized. To ensure these two directives don't cross wires, the cell physically separates the proteins into completely different domains. When nutrient signals are strong, AKT attaches a phosphate group to FOXO3. 14-3-3 chaperone proteins recognize these new sites and immediately latch on. Acting as biological tethers, they drag the FOXO3 complex away from the nuclear membrane, permanently sequestering it in the cytoplasm. With FOXO3 trapped outside, the nucleus is left wide open. Beta-catenin moves freely and docks perfectly with TCF or left complexes on the cell's DNA. This unobstructed connection activates the want pathway, leading to a cycle of continuous, unchecked cellular replication. By actively suppressing its own stress defenses in favor of proliferation, the cell achieves maximum growth, but leaves itself highly vulnerable to sudden environmental shocks. That vulnerability is exposed the moment a cell encounters severe metabolic stress, such as a sudden influx of highly destructive reactive oxygen species, or ROS. This sudden oxidative shock instantly disables the standard PI3K and AKT signaling networks that usually promote growth. Without AKT actively maintaining those phosphate groups, the 14-3-3 chaperones lose their grip and detach, leaving the FOXO3 structure unbound and active. Freed from its cytoplasmic anchor, FOXO3 executes a rapid translocation and aggressively drives itself straight through the nuclear membrane barrier. At the exact same time, the incoming reactive oxygen species activate stress-responsive kinases. This forces a surge of additional beta-catenin to accumulate inside the nucleus. Now both proteins occupy the same nuclear space amid high oxidative stress, an environment that rapidly induces post-translational modifications, or PTMs. These subtle chemical alterations restructure both proteins, radically increasing their binding affinity for one another. The result is a profound functional pivot in cellular decision-making. Instead of operating in isolation, the two proteins fuse into a single beta-catenin and FOXO3 macromolecular complex. In an instant, the cell has taken its primary engine for proliferation and forcefully recruited it as a dedicated partner for its environmental stress sensor. Forming this complex poses a mechanical question. How does joining with FOXO3 actually stop the WNT-driven proliferation that was already in progress? It works through a mechanism called competitive structural sequestration. As FOXO3 becomes active, it physically pries beta-catenin away from the TCF and left binding sites, leaving the TCF structure entirely isolated. We have concrete evidence for this process from co-immunoprecipitation experiments conducted on human colon carcinoma cell lines, specifically DLD1, LS174, and DL23. Researchers measured this exact interaction. This graph shows increasing FOXO3 levels along the x-axis and the amount of beta-catenin bound to TCF4 on the y-axis. Look at the trajectory. As FOXO3 activity increases in the cell, the amount of beta-catenin associated with TCF4 precipitously drops. The data proves that cellular transcription operates as a strict zero-sum game. For the stress response pathway to live, the growth pathway must literally be starved of its resources. By physically depriving TCF of its crucial coactivator, WNT-driven proliferation is utterly suppressed. Now in complete control of the beta-catenin payload, the complex navigates the genome, searching for specific FOXO consensus DNA binding sequences. It zeroes in on a very precise layout of base pairs, TT, GT, TT, AC. Docking directly at this exact sequence, the complex drives the immediate expression of the target gene, P27. The induction of P27 forces a strict G1 cell cycle arrest. The cell initiates a vital, protective pause, ensuring it doesn't accidentally replicate while its DNA is actively compromised by oxidative stress. Hitting the brakes via P27 is only half the survival strategy. The cell still has to actively eliminate the initial metabolic threat. While docked at the FOXO consensus sequence, beta-catenin undergoes a radical shift in its job description, shifting to a powerful transcriptional coactivator for the stress response. Together, the complex functions as an enhanceosome, a macromolecular machine that massively upregulates the exact genes needed for survival. The primary target is the SOD2 gene. This instructs the cell to manufacture the enzyme manganese superoxide dismutase, or MMSOD. Right alongside SOD2, the enhanceosome drives the massive parallel production of the enzyme catalase. These newly synthesized enzymes are deployed as molecular scavengers. They intercept the incoming jagged reactive oxygen species and rapidly neutralize them on contact. The sheer speed and scale of this biochemical reprogramming allow the cell to actively disarm the very oxidative molecules that triggered the alarm in the first place. By redirecting beta-catenin, the cell sacrifices its immediate growth to buy critical time, actively clearing lethal threats before resuming normal functions. This entire defense protocol raises a final question. What happens once the detoxifying enzymes do their job and the oxidative stress finally subsides? The formation of the beta-catenin and FOXO3 complex is not a permanent state. It is inherently temporary and designed to be fully reversible. When conditions improve, the complex dissociates. FOXO3 is silenced, and beta-catenin travels back across the nucleus to rebind with the TCF and LEF structures, safely restarting the cell cycle. In healthy tissues like renal proximal tubules, this process acts as an evolutionary rheostat. It is a highly sensitive, protective dial calibrated to ensure cellular survival during acute injury. But that exact same survival mechanism operates very differently inside the highly dysregulated microenvironment of a solid tumor. Cancers actively hijack this protective enhanceosome to survive the hostile, oxygen-starved conditions they create for themselves. By exploiting this pathway, a malignant cell can drive aggressive cellular scattering, develop profound hormone resistance, and ultimately fuel lethal metastatic progression. This is exactly why modern pharmacological agents, including targeted PI3K inhibitors and drugs like metformin, are designed to deliberately manipulate this specific signaling axis. They demonstrate we don't necessarily have to mutate the Wnt pathway itself to stop cancerous growth. We merely have to shift the structural balance of power between these two proteins. By mastering the contextual triggers of the beta-catenin and FOXO3 switch, we gain the ability to control this mechanism manually, shutting down pathology in oncology or activating cellular defense in regenerative medicine.